Hi, all of you, and welcome to resting membrane potential. That's it. So we're going to differentiate, right? When the potential is a resting one and when it is an action potential in a neuron. But the first thing we want to look at is a little bit about electricity. Electricity, we're always talking about positive and negative charges. And here we have some lightning showing you charge. It's in lots of different areas, but what you need to know as a big picture is that it's an electrostatic force and that like charges repel, right? And that opposite charges attract. And that's going to be really important in the body when we're talking about positive ions and negative ions and what they're doing. So work, right? Achieving something in these cells, work, can be done by moving charge. So moving charge is current. It's an electric current. It's like moving electrons along a wire that comes to your lamp. So it's the same thing. So we're always talking about ions when we're talking about charge and current in neurons. And it's measured in amperes, but we're not going to look at that very much. It's milliamperes because they're so tiny in the body. So let's think of it like this. This is a picture for you again, and here's a membrane, right? And there's something happening on the outside of the cell, extracellular, and there's something happening inside the cell, and it's quite different. There's a separated charge, right? And that is going to be potential to do work. It's like a dam, right? If you build a dam for water and you have all this water piling up, and there's going to be a lot of force there, right? If you make an opening, it's going to come out with a lot of force. So we're separating this charge, and it's going to have potential to do work. So it's called a potential difference, right? No water on one side, yes, a lot on the other. There's a lot of force on one side. And that's measured in volts, millivolts. But the first thing is, let's look at this picture more closely. Excuse me. So inside, intracellular, we have a lot of, let's look at these guys. A here, a lot of these proteins. And these proteins are negative. Lots of negative stuff going on. And then lots of these potassium ions that are positive. A few little chlorine and one little sodium guy. If you compare that to the other side, very different in the quantities of those ions. On the outside of the cell, many sodium, lots of chlorine, and a tiny bit of potassium. So just think of it as, again, the dam, right? That there's potential for something to move from one side to the other, in this case, and from the other to the one, right? Because there's a concentration gradient of these guys. So if you allow them, they're going to move. If you open the dam, it's going to go. Okay, so lots of different animal cells use electrical signals, and actually plants do too. This is aloe vera, and they found out that they have signals, which is amazing. And I don't want to go into what that does, but it's during heat. So when it overheats, it produces charge. The heart, this is a picture of the heart here, that needs these electrical signals to be able to send a message to the muscle for it to contract. Loved up, loved up. So lots of electrical signals everywhere, and we can measure this. We can measure the charge, right? So the membrane potential, that word again, potential, is the difference in charge between the inside of the cell and the outside. But we're always going to measure it in terms, which are we measuring? Are we measuring what it is outside, or are we measuring what it is inside? We're measuring what it is inside because we're talking about cells. And it's measured in millivolts, thousands of a volt, okay? Very tiny, but makes a big difference. So how is this potential created? So for example, in this picture here, there is electrical equilibrium, because if you count all the positive ions and all the negative ones, it's the same in and out. Nothing's happening, right? It's no difference in the water from one side of the dam to the other. Here, however, there is a change. There's a gradient formed, and we have, let's see, this movement through ATP is moving some of these positive ions out, right? So if I could take some of that inside the cell and then put it out, kind of like the dam, and keep it outside, then you form a gradient. And then if you allow the opening, that's going to flow back in again. Okay, so we create a gradient, and that's exactly what cells do. They create a gradient for themselves by pumping some of these ions out. So, which human neurons have high intracellular concentration of potassium, like that drawing showed you. Lots of potassium inside. We keep it that way by pumping the sodium out. So it's low intracellular sodium. And if you keep just pumping it out and keep pumping the potassium in, then you're going to end up with that situation where there is this potential difference. Like the dam, you had to build something really strong for it not to go in the other direction. So this is it. We're putting energy into creating that difference. Inside the cell, on top of the sodium and the potassium, there are these large proteins, hundreds of proteins, and they are all negative. They're very negative, very super negative. Okay, so if I take a voltmeter and put it inside a cell and compare it to the outside, it's like, oh my, very negative. Why? Not because of potassium that's positive, not because of sodium that's positive. It's the proteins. This was studied first in the giant squid. So it turns out that they have very large axons. Not only are they a big animal, but the axons themselves are actually quite large for the size of the animal. So they can be, in these big guys, it can be the size of spaghetti. So you could put that under a microscope and really put in a little probe inside and out and find out if there's charge there. If at a resting potential, it's true what I'm telling you, that it's more negative inside than out. Well, that is done. And all it takes is a probe. So if you had a cell in saline, and it takes a probe inside the cell and another probe on the outside where it's bathed in, and you would want to see, gosh, I can get my face out of everything here. You'd want to see with a voltmeter, like you would if you go in the outlet, do I get a reading? Is there truly a difference from inside to out? And if you do that in a cell that is resting membrane potential, you're going to get a reading in millivolts, thousands of a volt, of about approximately negative 70 millivolts. And that'll differ in some of the different cells. So the resting membrane potential needs to be maintained, right? Because if you just let the water flow out of the dam, it's just going to flow and it's over. So you need to have that strength to put it there. So it's maintained by a sodium-potassium pump, and that takes ATP. So it takes force. It takes force to get that wall up, to keep that water in the dam. And ATP, in this picture, it's showing you what it does. It's going to take those sodiums that came in out, because we want more of them out, and it's going to take those potassiums that went out, we're going to put them back in. So it's essentially fixing what happened previously. We're going to want to maintain it, that when it's resting, it's going to be the minus 70. So this transporter, which is a protein transporter that's on the membrane, will transport. For every 3 sodium ions out, it brings 2 in. And it's this constant pumping. It's kind of like the pump that you would have if you had a basement and it flooded. You would have to, that's more like on the East Coast, right? You have these houses that have basements and it gets very wet during the rainy season, and you have a pump that clicks on automatically and it'll just pump some of that water out. It's hard to get water from below the surface level up. The only way you can do it is by pumping it with a pump, which in this case takes ATP, takes force. So it keeps transporting those sodium ions back out and the potassium back in. That's cool, this little guy. What if the cell is not resting? Resting, minus 70. Cool. Then something must change in the membrane. Minus 70 isn't maintained. Some change happens. What is going to happen for that not to stay stable like that? What's going to change? We kind of open the dam, right? And so that membrane permeability to sodium changes. In other words, the dam is no longer a barrier to that water, that permeability to sodium. So if I have a lot of sodium outside the cell and all of a sudden I open channels and say, well, okay, then it's going to flow in, correct? So by flowing in, it's going to go here on the membrane potential from -70. And inside it was -70. So all of a sudden you've got these positive ions. It's going to climb and then come back down again. And we'll see that in more detail in the following video, where exactly we look at what's happening at every point in time. But for now, depolarize gets positive, repolarize back to negative, depolarize positive, repolarize back to negative. What if I overdo it and do too much? It hyperpolarizes that membrane. So it happened how again? It happened because there was an increase in the permeability to sodium. In other words, the channels that used to be closed to sodium, like this, are now opened. It's like, okay, some signal came, some signal to that cell, because it has to act, right? Some signal is coming to it saying, okay, open those channels, and in flows the sodium. So depolarization is an increased membrane permeability to sodium in a particular channel. Closed, closed, open. So when that signal comes, it comes both to the sodium channel and to the potassium channel. So they should both open at the same time, but potassium is a slowpoke channel. And once it gets a signal, it has to decide still, take a while to slowly start opening. By then, sodium's already run away with the show. So it's already depolarized. And by the time potassium has the time to react, it's a repolarization that's happening. It's returning the membrane to resting membrane potential. So once you open the potassium, the sodium starts to flow out. And it goes back to -70. So -70, -60, -50, -40, 0, +30. And then back down, +20, 0, and all the way back down to -70, overshooting a little bit and then coming back up. That was repolarization. This is my next slide. I got ahead of myself. What if too much flows out? Then it's called hyperpolarization, and then it drops below -70 millivolts. And it'll slowly come back up again. Okay. Some anesthetics work that way. They drop really low, -90. And so if they go really low, then it's hard to stimulate that nerve, that neuron. And if you can't stimulate those neurons in a nerve, then you don't feel pain. Oh, very cool. So what changes the permeability of the membrane? Current, voltage, hormones, that would be chemical, stretch or pressure, just touching something is going to distort it and open a channel, heat or cold, and I'll repeat that over again in one of the future slides. So all that to tell you about those two master players, which are sodium and potassium. Okay, thanks guys.